Okay, today I'll be talking on pathogenicity islands and their role in the bacterial virulence and survival. So as we discussed in my earlier lecture about the pathogenicity and the virulence factors of the microorganisms, especially in bacteria and viruses, these factors are responsible uh, for facilitating the pathogenicity or for promoting the bacterial or viral particles or organisms to be more pathogenic and more infectious, right, and more contagious to establish a disease or infection. Now, having known that if the virulence factors are suppressed, then the disease or pathogenicity or infection level may also be reduced or controlled. So, what is the reason behind that, and what is the mode of action and mechanism behind these virulence factor expression or presence of virulence factors, or the transfer of the virulence factors from one bacteria to another bacteria, right? All these things to be discussed today in light with the pathogenicity islands and its role in bacterial virulence and survival. Now, if you look at the bacterial genome structure, it's a typical bacterial genome structure consists of two main components. We call core gene pools and flexible gene pools. Now, what is the core gene pool? Core gene pool is comprised of essential structural features and fundamental metabolic pathways which are responsible for the survival of the growth and survival of the survival of the microorganisms, survival of the bacteria. They are characterized by the relative homogeneous GC content and are normally encoded in a stable region of the chromosome. A stable region of the chromosome means like chromosomal region which are continuously perpetuate to cell transmitting their genetic material, which are conserved in closely related species. For example, if you talk about the Escherichia coli or Klebsiella pneumonia, so these regions are very conserved and they remain constant in every species when it passes through vertical transfer. Whereas the flexible gene pool is something which is may called variable and encoded by the functional and only advantageous under the specific growth conditions. For example, if the cells are growing under a specific condition, they can adapt some conditions, so such kind of a region of a genome is activated. It comprises variable regions of the chromosomes and mobile elements, IS elements, which can move from within the chromosomes as well as from one bacteria or one microorganisms to another microorganisms. So these IS elements or TN elements are highly mobile genetic elements, right? And these mobile genetic elements are moved from one strain to another strain within the species and across the species even through conjugative transposons and through integrons and superintegrons. Now these integrons and superintegrons are the part of, they are not carrying the essential gene, but they are basically associated with certain gene which can move from one organisms to another organisms. So there's a difference between these two, that is a core gene pool and flexible gene pool, is that in core gene pool they are always constant, always conserved within that species, right, and they move through one bacteria or one microorganisms to other microorganisms through the vertical transformation, through the vertical transfer, mode of transfer. Whereas the flexible gene pool could have also transferred through horizontal gene transfer if they acquired by the plasmids in the cell through recombination process. So many genes encoded the toxin, adhesin, secretion system, invasive invasions and other virulence factors are virulence associated factors that have been found and encoded by the mobile genetic elements. They've been found to encoded with the IS element, integrons, and the conserved chromosome backbone is interspaced through these sequences with the large regions of the exhibit features of former mobile genetic elements and they form genomic islands. So this is called, this region, these genetic elements termed as the genomic island, GEI. Now the concept of genomic island is basically can be explained here. The features of the genomic islands, what are the features of the genomic islands and what are the attributes and characteristic features of the genomic island. So GEI are the genomic islands are present in many bacteria, but absent from closely related strain or species. So they are present in a number of bacterial populations, but they're absent from closely related strains. For example, within the E. coli, if you have very closely related strains of the same MLST type or same type of strain, same type of species, then probably they may not present in every same type. And this genetic mobile elements are genomic islands are basically occupy the relatively large regions of the chromosomes, up to 10 to 100 base pair. Because they are present, mostly they are present sometime on the flexible region, and GI differ in GC content and codon region from conserved regions of the chromosome. So the conserved region of the chromosome is slightly different from GC content and codon regions as compared to the flexible regions where the genomic islands are present. Around we call them as a pathogenic island and pathogenicity island. Now genomic islands are often flanked by direct repeats. So the concept of direct repeat and the indirect repeat is that, so this is how we can see, suppose this is a region of the gene, and here these are the, so these genes are flanked by these direct repeat 1 and 2. Now these direct repeats are basically flanked by, this gene is flanked by these direct repeat. Now this gene, for example, ABG. Now this AB gene, when it is flanked by direct repeat, it can be either deleted or inserted through recombination process. Now when you take a direct repeat, these tandem repeats, which are palindromic sequences present on both the sides, one is on the right and left side of the gene, or if gene are associated with this direct repeat, then these genes either go through deletion process, it can be deleted from this, or this can be or another gene can be inserted here. So this insertion and deletion process is governed by the direct repeat. Now if you look the inverted repeats, which are not present in this case, then the process of inverted repeat let the gene inverted. Now this inverted means like inversion takes place, and this inversion basically involved in the gene expression process, which we discuss in some other time. Now let's see how genomic elements are involved in this process of gene transfer, and the transfer and associated gene transfer from one species or a strain of one species to another strain of a species. So GEI, that is genomic elements, are often flanked by direct repeats generated during the gene integration, and this integration process will either through deletion or through insertion process, as I have just talked about. Now genomic elements are frequently associated with the tRNA loci, and which are preferred target sites for the integration process. All right. Now GEI often possesses the functional or cryptic genes for genetic mobile elements, which integrase and transposon. So here we can talk about how the integrase and transposases, which are the enzymes carried by these IS elements, are carried by these mobile elements, which helps into deletion or integration of these genes in the genome of the bacterial chromosome. So these islands are basically functional islands, are basically help in mobility, or they can move from one genome of the bacteria to another genome through these integrase, support of the integrase and transposase enzymes, which help in through transposons and integrons. Now some of the genomic elements tend to be unstable DNA regions due to recombination between the flanking direct repeats, and these GI are prevalently found in the organisms that show the frequent gene transfer by bacteriophages and plasmid, which are regarded as a possible precursor of GEI. Now here the role of bacteriophages is also important, because we will talk about the transduction process also when we maybe in the next topic. In the transduction we can have the bacterial genome, that is the bacterial genome is being integrated through undergo lysogenic process of the phage after the phage infection, and then it can taken away from one bacteria to another bacteria after integration with that. So this kind of a things mobility is basically very prevalent in this process of transformations and transduction process of, and make the new species through variations and through new mutations, and new variations are formed through these processes. Now, GI contributes to bacterial fitness. All right. How does GI, that is genomic elements, contribute to bacterial fitness? GI basically often carries to determine that they're responsible for various environmental changes in the host bacterium while the genes are transferred from one bacteria to another bacteria, thereby increasing the bacterial fitness and survival. All right. So it can also produce the some protein carried by the gene which are transferred through these elements, and they may also responsible for the fitness of the bacteria. Fitness here means like bacteria can have a strong defense mechanism, strong virulence factors which can fight against the host system in the environment, and they can survive for a long time. Otherwise, the bacteria may be killed. If the bacteria is, for example, I can take an example of capsulated pneumococcus pneumonia and non-capsulated pneumococcus pneumonia. So the capsulated pneumococcus pneumonia is very strong because they have a capsule, polysaccharide capsules, and they can have they cannot be phagocytosed by the defense system of the bacteria of the animal, and when injected in that, it may cause disease. Whereas on the other hand, if it is non-capsulated, which are smooth, not rough, and these non-capsulated will basically responsible for they were not responsible for the death or the disease of the animal because they have been phagocytosed by the internal defense system of the animal. So this is how it differ from, this is how it allows the bacteria to survive. And they also increase the life cycle of the bacterial population or bacterial species. Now, pathogenicity island, which are also part of it, because under the genomic island they've been divided by the pathogenicity island, ecological island, resistant island, and symbiosis island. So all these four categories are the subset of the genomic island, which has present in the genome in different categories and have different function. So pathogenicity island are we call PAI, are basically encoded by the virulence state, such as a toxin, addicin, fimbriae, some factors for the host cell entry, some capsule secretion system, iron uptake system, and so on. So if these genes which are responsible for fimbriae formations, pili formations, some addicin like AG1, AG2 in case of S mutants and some gram-positive bacteria, if these genes are carrying these factors and they are associated with the particular region of the chromosome, and they are responsible for the expression of the virulence factor in the microorganisms and the bacteria, we call that region we call the pathogenicity islands. Whereas the ecological islands are contributing to adaptation, to specific growth. So these are some genes which are responsible for providing some support for the bacteria or for the microorganisms to survive in some different environment. So they have basically used for adaptation process of the different environmental conditions, such as the temperature, pressure, humidity, right? Sometimes pH, salt. So all these things can be adapted by the microorganisms in different ecological conditions, and the genes that are responsible for that are the regions which are present on the chromosomes. The gene region present on the chromosomes are responsible. So this region of the chromosome or the gene is called ecological island, such as iron uptake, sucrose metabolism. Then we have a degradation of the phenolic compounds. Sometimes phenolic compounds may be toxic for the cells. They cannot survive in the presence of phenol. So what they do, they basically have some gene which can express some enzyme which can reduce or degrade the phenolic compounds or phenolic compound derivatives so that they can survive. So basically the bottom line is that all these islands, they are supportive to the microorganisms for their survival and for the growth and for the long life. Similarly, we have a island, resistance RAI. Resistance island basically against it enhances survival by the providing the resistance. So various drugs, various antibiotics such as MEC in locus in case of Staphylococcus aureus, and we have some other genes, intrinsic gene like in Pseudomonas aeruginosa. Several antibiotic resistant markers are present, and these markers are basically now. So we are not talking about here is about the plasmids which are acquired by these bacteria for survival, for adaptation conditions, or for adaptation under certain environment, under certain harsh condition, or in the presence of the enemies like a drug, antibiotics and other harsh conditions. No, it is basically the part of the genome, so we call them as an island. These regions are already present in the genome. Similarly, symbiosis islands are they carry the genes which provide for the symbiotic relationship, such as the nitrogen fixation and mesorhizobium in case of mesorhizobium species. And now these regions are basically allow these bacteria to survive in the roots nodules of the rhizobium plants, nitrogen leguminous plant, and they provide the nitrogen. They fix the nitrogen for the plant. So they have the food from the plant in the during the nitrogen fixation, as well as they provide the nitrogen from the atmosphere and they provide to the plant. So plants are benefited, and these bacteria are also benefited. So when the two organisms or two individuals benefited each other, and for the survival, they are called the symbiosis relationship. And this relationship, symbiotic relationship, is governed by certain regions in the genome of the bacteria we call the islands, and these islands are called symbiosis islands. Now some genomic islands have been assigned to different subtypes depending on the habitat, genetics, background, and their respective bacterium, such as high pathogenicity island. Now if you look into the whole genomic island, including the pathogenicity island, ecological island, symbiosis island, and resistant island, all these kind of regions rather which are present on the chromosomes are supporting the bacteria to survive, right? And these regions are also responsible for associated with some mobile elements, integron, transposons, and others. So they can also carry from one place to another place time to time. Now PIs of the enterobacterial pathogens are basically important, and this pathogenicity islands in enterobacterial pathogens are important because these enterobacterial bacteria species of bacteria from this family of bacteria, they are pathogens, and if these islands are expressed in the bacteria, the pathogenicity is increased, and so we call the pathogenicity island. Now the most characterized genomic islands have been found in the member of the Enterobacterial, or now we call Enterobacterial. Earlier it was called as Enterobacteriaceae family, indicating the pathogenicity islands have play a very important role in evolution and of Enterobacterial pathogen. Escherichia coli normally lives in harmless commensal, but some variants can cause diverse infections like uropathogen. So the same E. coli, which is a non-pathogens, has become pathogenic after having the expression of these pathogenic island or pathogenicity island in the bacteria. Gastrointestinal diseases like urinary tract infection and systemic infection. In gastrointestinal diseases like ranging from cholera, like diarrhea, to severe dysentery, and some hemorrhagic colitis. Sometimes the blood comes in the stool. So this kind of infection is caused by these bacteria, which are coliform bacteria, even E. coli, which are having these islands, because these islands provide the bacteria for surviving the host system and does not allow the bacteria to be phagocytosed or combat with the defense system of the immunity of the host. Now UTI infection is very common in many bacteria, like uropathogens like E. coli and Klebsiella. Both they may carry various markers and various islands, various regions on the chromosomes which provide the support for the survival even in the urinary tract or epithelial cell lining of the urethra and urinary tract, basically a genital or region. It includes the cystitis and pyelonephritis. Sometimes it may lead to severe infections like cystitis and pyelonephritis. Also, in case of kidney failures, may also be takes place. Systemic infection are very common, having these bacteria with the pathogenic island, providing the high virulence factors, which are very stable, and under the mobile elements can also shift these mobile elements through these mobile elements from one bacteria to another bacteria, and they cause the population of bacteria in a region to be infected by pathogenic state or pathogenicity, such as septicemia and meningitis. The very common disorders, very common diseases and infection which may lead to even death of a person, death of a patient. So pathogenic E. coli, which may cause the UTI infection, right? So classified into different pathotype. We call UPEC, EPEC, EHEC, right? So urinary pathogenic Escherichia coli. Then we have and Enterobacter pathogenic Escherichia coli, and then we have EHEC, etc. Now these different type of E. coli are basically generated, are developed through these pathogenic island, because if you look the E. coli, it can have thousands of different strains of E. coli. Now these E. coli strains are divided into different strains based on these pathogenic island. If you talk about in light with the pathogenicity of the bacteria, whether so one strain may be pathogenic and the strain may not be pathogenic, right? So what make the strain pathogenic is basically PI elements. So these are characterized by specific virulence factors that capable of causing the high infectious rate, and pathogenicity conditions are make more pathogenic and exploit new niches in their host, and they more cause infection in the host, and don't allow host to evade it because of the either weak immune system or their strong virulence factors. So many of these key virulence factors are included by these pathogenic island. So it is not a single virulence factor, but I think it is a multiple virulence factors are basically responsible for transferring these things. Now example of cell C associated with the PI. So example of cell C is associated with the PAI, pathogenic island, and this pathogenic island is basically, cell C is a very specific pathogenic island present in bacteria, and these the organization of this, if you look into this figure, I think this is very clear that F17 like fimri which are present. So this F17 like fimri is basically present on the PI, that is pathogenic island, CS12 like fimri, then alpha hemolysin. Now these regions which are present on this genome are pathogenic island. Now cell C is basically is responsible for the integration of these regions from one bacteria to another bacteria through mobile elements, and these regions are responsible for the virulence factors. Now similarly you have cell C secreted by another gene like TIME3 secretion system, then we have a protease, then we have addison. So all these factors, that is addison factors, which help in the addition of these bacteria on the surface of the host, maybe epithelial cell lining, or respiratory tract, or urinary tract infections. So basically if you block these addison, it can reduce the addison process. So to know these virulence factors and to know these pathogenic island, that is what are the region of the chromosome which are responsible for the pathogenicity. Similarly, this is called arobactin synthesis, and this is called survival in macrophages. So cell C is responsible for the survival in the macrophages, because macrophages are the cell in the host where they involve in the immune system to evade or to engulf or to kill the bacteria through their attack. Now because these are just like army, and these army attack on to the invaders, right? And when the army is strong, then invader cannot come, but if invader is strong, so in this case these pathogenic island or these factors are basically responsible to make the pathogen more stronger, invader more stronger, so the army cannot work. They can even they can fight with the army, and they can win to the army, and that's how the infection is established. So this is very interesting to understand the whole sequence analysis of cell C associated PI. This is the example of enterobacterial family member where the CP found. Now, of operating frame with the similarity to transposase are indicated by the hatch arrows, like these arrows, and these SPI3 for Serratia and Enterica bacteria present, and highly similar gene for the present in all island. So these are some specific example of some specific bacteria, but I think it is present in many of the bacteria. These systems are present, and since these systems are associated with the transposon, these systems are associated with the mobile genetic elements, so they can easily move from one place to another place, right? And that's how it can difficult to control the infection having these bacteria with these islands. Now, whereas most PI, that is most pathogenic island in pathogenic E. coli shows that like mosaic-like modular structures, right? Whereas in certain cases, these pathogenic islands are show similarities in the presence and linkage of a certain virulence determinant. Now, because there are certain virulence determinants are also present, which determine the virulence factors or their expression of the virulence factor into the bacteria, and they also allow the mobility of these virulence factors from one bacteria to another bacteria, right? And this may also cause the variability among the population of a bacteria, right? Now, variability here means like suppose if you have a E. coli strain and the single E. coli strain may vary from other E. coli strains, right? It is because of the mobility of certain island or certain gene of that islands. Say for example, in this case, not necessarily with that all the component of the cell C system or all the component of this pathogenic island can be transferred from one bacteria. So in say for example, in one of the bacteria you may have this kind of a system, right? But in another bacteria you may not have this. You may have some, you may not have aerobacterian synthase, you may not have adhesin, you may have some biofilm forming genes. In other you may not have biofilm forming genes. Similarly, you may have in certain cases you have some quorum sensing gene, in other you may not have a quorum sensing gene. So this is all this is all being controlled and governed by the presence of the PAI, that is a pathogenic island, as one of the component of the genomic island. So in a summary, we can explain in this lecture that what is the role of a genomic island and what is the role of pathogenic island, that as a part of the genomic island, that it provides the sustainability, it provides the resistance against many immune responses of the host while it infect. So basically when the PI elements are there and they also transfer through transposons and genetic element, so they can make a number of bacteria in a particular site. For example, if you talk about the gut, if they have some pathogenic bacteria present in the gut, and they may somehow through feces go through the urinary tract infection, and then in the urinary tract infection in women it is very common because of the anatomical structure of genital organ. Now when it has become very common, then it causes the recurrence of the UTI infection, and then they have the bacteria make a niche in the particular site of urethra, urinary tract, the whole urinary tract, urethra. Now when it does, then these bacteria can have a capacity or capability of transferring these PAI, that is a pathogenic island, or some of the genes present on the pathogenic island responsible for the virulence factors expression, responsible for the pathogenicity of the bacteria. So when they keep transferring and the bacteria keep growing in this particular region, the infection chances will increase, and you may have sometimes recurrence of the UTI infections again and again. Now this is all about today's on this topic, but the detail of this PIE and the role in the uropathogens will be discussed in the next talk. So I think this is all about for this genomic islands and virulence factors and pathogenicity associated with these genomic islands, especially pathogenic island. Thank you very much.